Good afternoon, ladies and gentlemen, and welcome to another exciting episode of Scientist Saturdays with the Cleveland Museum of Natural History at home. If you were watching on Facebook looking for us, sorry, we had a little glitch there, but we're pretty sure we've got it under control. My name's Lee, and I'm in charge of video conferencing content for schools at the museum. But today, I get to address one of the scientists who does some pretty cool research, Dr. Gavin Svenson. And I want to make sure I get your title completely right, so I'm going to read it off my screen. Dr. Gavin Svenson, Director of Research and Collections and the Curator of Invertebrate Zoology. Good afternoon, Gavin. How are you doing this fine Saturday?
I'm good. How are you, Lee?
Feeling great. It's a nice day. Going to get outside and maybe look at some insects later on, since that's what we're talking about today. And let's clarify for our viewers precisely what is the field of invertebrate zoology.
Well, invertebrate zoology is the study of all animals that don't have a spine. And that actually is the vast majority of animal life out there. Some figures are 95% of animal life are invertebrates. So if you want to study biodiversity, that's the place to be because there's a lot to look at. And at the Cleveland Museum of Natural History, most of our invertebrate zoology collection consists of insect specimens. So that's the study of entomology. And we also have some freshwater mussels, but most of us, including the other curator that work at the museum, are entomologists and working with insects.
I did have the chance to interview Dr. Nicole Gunter, I think it was 2 weeks ago, and she was talking about her specialization in dung beetles and that she had devised some sort of nifty little yellow pan trap full of water to capture specimens around her home while she's been in the quarantine times. I don't suppose with you, praying mantises probably would not be too interested in a yellow pan?
No, they're not. It's a lot of insects come to a lot of different colors, and they're queuing in on flower or pollination. And these yellow pan traps seem to just mimic that just right. And it's not any yellow pan trap; it's a certain yellow pan that the insects respond to the best. And you just put soapy water in it, and you can catch a lot of insects like that. And mantises, that's my specialty. They don't really come to that at all. So I've never really used that. And what I've been doing when I'm at home. During the quarantine and stay at home is look at birds because I haven't had that opportunity to look out the window every day. So now I am.
Well, that's fun because it shows that scientists who specialize in one area still maintain interests in others. And man, working at a museum, hey, you get to interface with all of those. Well, since we're not talking about a praying mantis from Ohio, I guess we'd better travel, and you get to travel a lot of awesome places. In the video we're about to watch, where are we going?
We're going to go to the northern part of Peru on the Amazon River. So the Amazon extends all the way through Brazil, all the way up into northern Peru, and there's a huge swath of biodiversity that lives along that river.
Fantastic. I'm ready for a trip. Hey, show director Matt, take us to Peru.
I'm Dr. Gavin Svenson. I'm the Curator of Invertebrate Zoology and the Director of Research and Collections here at the Cleveland Museum of Natural History. Back in 2013, we were on a museum expedition to Peru. And we've gone to Peru in the past, but this was a special expedition in the northern part. So we actually were right on the Amazon River. And we went to a research site that looked pretty promising based on the habitat there. We set up a very large light trap, which attracts insects, kind of like your porch light on a house attracts insects. We waited a little bit, and there were a lot of other insects coming in. A lot of them were beetles, wasps, flies, other species of praying mantis. And then this tiny little thing came in that was bright orange with a little black head and a little black abdomen. And it ran around on the sheet, and it looked just like a little wasp. I knew it was slightly different. It didn't quite look right. It didn't look quite like a mantis. It didn't look quite like a wasp. I went up and I grabbed it and I put it in a little vial. And then once we started observing it, we knew we had something totally different. We assumed that bright coloration was a mimicry of something because if you're defenseless and you're depending on mimicry, and the vast majority of other praying mantises are mimicking tree bark or leaves or blending in to avoid detection, if you're conspicuously colored, you're advertising something. Not only does it have this black, orange-black coloration pattern that looks like all of these wasps, but behaviorally it moves around like them. It rotates around fast. Its antennae are moving very rapidly, like a lot of wasps will move their antennae. And it also does this abdominal pumping behavior, which is very characteristic of a wasp. So the mantis is really just cueing in on a signal already present in the environment as a visual defense to be left alone. When we got it back to the lab and actually did the research on it, we conducted internal dissections, and that confirms that we have an independent character set that really tells us that this thing is unique. And we directly compare it with hundreds of other individuals in the lineage to also confirm that it's unique. It actually matched quite well with an already described species. Now, our species came from northern Peru, and this other one, which looked very, very similar, but it was not brightly colored, came from French Guiana. But when we did the internal dissections and all the measurements, and we looked at the other important characteristics that defined what our new species looked like, it had similar characteristics. They weren't similar enough to be the same species, but what it really told us that we have a new lineage. So I can tell you, Vespa mantoida, which is a name we got it, wasp mantis, because of this wasp mimicry. It really is telling you that these two species within that lineage are uniquely characteristic of this particular biology. Every time you find something new, it gives you the opportunity to reevaluate what's already known within a broader lineage. Most species discovery doesn't happen right in the field like that. You don't just see something and say, Eureka, that's a new species. You usually have to come back and you put it under a microscope, you do all of the analyses, but in these rare moments when you're out in the field and you see something like that and you know exactly that it's different, it's really exciting.
Wait a minute, that little tiny mantis looks nothing like what I visualize when I see a praying mantis here in Ohio. Plus, I wish they had some footage of you just standing at the trap going, Eureka, because that would be awesome. But that would be a great moment.
If you saw me at the trap, I would have been highly stressed out, because once I realized what I was looking at, then I had to actually catch it so I could observe it and then confirm what I was looking at. Because oftentimes I've been in the field and things, really, really amazing things have flown into a trap, sat there, allowed me to see them just for a split second, and then they're poof, and they're gone. So catching it is half the battle there.
Seriously, and that was a really little guy too, so I would imagine that he or she is pretty doggone fast. But because I'm pretty sure that anybody watching that video that lives here in Ohio is going, whoa, because the mantises that I normally, like when I was a kid catching them in my backyard, because I think they're amazing, they were those huge green ones. And it wasn't until I worked here that I found out that those are not even native to Ohio. So let's take a look just for a moment here at this amazing diversity of different types of mantids, if you don't mind.
Yeah, that one right there, that's a nymph of a plant mimicking mantis in a lineage that includes flower mimicking mantises, stick mimicking mantises, and even there's mimics that mimic a broken off stick. So the part of the stick that's still stuck to the other part of the branch, you break that stick off, there's a mantid that mimics that one thing. I don't know what to call it. We could probably coin a new term right now, but it's pretty camouflage. And yeah, you're right, Lee. The vast majority of mantises are really, really different than the ones that we have here in Ohio and frankly across most of the United States. So there's about 2,500 species of praying mantises worldwide. The vast majority of them are in the southern hemisphere. So that is from their evolutionary origins on a big supercontinent called Gondwana. And the ones that we have here in Ohio, the Asian mantis, the Tenodera sinensis, was imported into the US probably around 1900 in the eastern US, and it was used as a biocontrol agent. And a biocontrol agent is an insect that you want to feed on the pest insects that you want to get rid of. But mantises, they're not good biocontrol agents because they eat everything that's good, they eat everything that's bad, they eat each other, and there's even documented cases where they eat hummingbirds. So they're not a great biocontrol agent. So, but that's what they were brought here. But they really reflect the standard green morphology that you see in a number of different groups. But really, the vast majority of them look nothing like that. And they have all sorts of cool specializations, like flower mimicry, bark mimicry, and like the one that you just showed a picture of. I think you have more pictures, don't you? Yeah, this is Coeuridotus. This is the common name for this is the shield mantis. It lives in Central and South America, and it mimics a living leaf. As you can see, it looks just like it, and it kind of hides on the surfaces of leaves. What makes mantises really neat is that they their camouflage is pretty much their own defense. They can defend themselves a little bit by striking back at something approaching them, but if they blend in, that's their best strategy from bird predators or lizard predators or other mammal predators. This is an orchid mantis right here. This is a nymph. Unlike the picture suggests, they don't actually live on orchids, but they live on green vegetation, and they do what's called masquerading crypsis. So they actually become the flower to attract pollinating insects like bees, and the bees fly right up to them, think that they're a flower, and then they nab them right out of the sky. So they're pretty convincing flower mimics. Well, this is the mantid with the best of names, Idolomantis diabolica. And this is a threat display or a defense display. So this is kind of one of the defense mechanisms that mantises have, and this coloration patterns. Maybe that's an eye spot or something like that. We're not really sure what it is, but if a mantis is found out in the environment or a monkey is approaching them, they can rear up and get themselves to look bigger and display some fancy colorations and try to scare off the predator that might be attacking them.
Now, I've seen a few videos of I think they were leaf insects that kind of rock their bodies back and forth as they're moving, sort of mimicking a leaf in the wind. And in the video, you mentioned how your new mantis is mimicking that pumping abdominal behavior of a wasp, which is fascinating because that's a whole secondary level of camouflage, moving your body. Let's talk about that a little bit, and why that little guy was so unique.
Yeah, this is a cool thing about a lot of insects when it comes to mimicry. You can mimic plants, which is quite common because you're trying to camouflage yourself and blend in. But this little mantis is unique among all other mantises because it's conspicuously colored. It's brightly colored, and it's brightly colored because we think it's mimicking a wasp. And black, orange, black coloration patterns are really, really common across a lot of different wasp groups. And this one seems to have cued in or evolved that same coloration pattern. But even more, it doesn't do the typical hide away behavior, but it wanders around openly on vegetation and behaves like a wasp. That movement pattern, that fast rotational movement pattern, that the movement of its antennae that go like this, and then its abdomen moving, all of those are things that are very characteristic of a wasp, kind of patrolling an environment looking for prey. And this mantis does all of those behaviors and then has all the visual characteristics to look like that, which makes it really different from most others.
The antenna is a fascinating part of it because I know the big green mantids that I would hold on my finger in the summer times, that I don't think I ever saw their antenna move. If they did, I was distracted by the giant gripping front legs. But man, that little guy really does twitch his antenna a lot. That's cool. And the fact that it's so
big
fast.
ones don't seem to do it.
Yeah, I want our viewers to really appreciate how this whole eureka moment, how tricky it is for you to cue in on a new insect behavior among all the ones that are being attracted to your light traps. And I do believe that show director Matt has a little bit of video. So let's take a look at what a light trap looks like in action, and you can explain what's going on.
Yeah, I would best describe it as a visual obsession. When I'm in the field, I think I cue in on the shape of a mantis, and it becomes kind of my search pattern. And I think a lot of people have search pattern capability, and especially ornithologists and people looking for fossils see these things very easily. And I'm doing the same kind of thing here. This is a sheet in Gabon, Africa. It's one of the best collecting locations that I've ever been to. And you can see this is a huge moth diversity. So a person studying moths would freak out when they looked at a sheet like this. But I was specifically looking for the mantises. And for me, it's actually really easy to look at a sheet like this and pick out the shape pattern that I'm specifically looking for. And a different biologist might stand here and see a totally different thing and cue in on a different shape pattern. So it's about training and familiarity and knowing what you're looking for. I've been in the field with people that study fossils, and I can't see it at all. And if those same people came with me into the field and looked for praying mantises, they wouldn't be able to see it. So I mean, some of the best times in the field that I've had is when I've gone with a herpetologist to study snakes or frogs, because all of a sudden they're finding all of these things, and normally I walk through the forest and I don't see most of them. So it's a neat thing to to work with other specialists. You can see some of the moths right there. That's a great one right there. It has these very, very long tails coming off of there. There was a friend of mine who's also an entomologist who studies moths, showed that the tails were for kind of the same thing as like a gecko tail breaking off. It's like a defense. When they're flying, they can defend themselves by having a tail as a break off mechanism. This is a really colorful metallic moth in the tiger moth family. But you can see it's just immense diversity out there. And that big light bulb right there, that's a what's called a metal halide light bulb, and it's giving off full spectrum light and a lot of actual ultraviolet light. So one of the questions we usually get are why do insects come to lights? And we don't have a specific answer for that. We can't tell you exactly why it is, because it's hard to get into the mind of an insect. But most insects see ultraviolet light, and what's cool at night, the moon reflects ultraviolet light much more than other spectrum of light. So if you're an insect and you're navigating at nighttime and you can see ultraviolet, you can actually see the environment quite well and navigate by the moon. So we think that
you had
light
mentioned
that gives off more ultraviolet will actually work better for attracting moths compared to a kind of a yellow porch light that a lot of people have.
Gotcha. Sorry about that. It's always that 2-second delay, man, in the transmission. You had mentioned the shape, training your eye to recognize the shape of a mantis. So I was trying to do that while the video was running, and I know I spotted at least 2, one up closer to the light and then one kind of in the middle. And I was getting all excited for a minute. And for our audience, just as a perspective, I think everyone does that to some extent, because when I was a little kid, one of my sources of pride was whenever my mom would lose an earring back, an old school earring back that had that double loop shape into our shag carpeting, I would always be able to find those things because I was looking for that very specific shape. So anyway, just something relatable for our audience. What does a praying mantis look like?
Lee, you'd make a good entomologist, I think.
Oh man, I just want to grab everything off that sheet. And man, that was diverse is the word. Like I want a word bigger than diverse to describe all the shapes that were going on that sheet. And it's a big part of what you're studying, right? Is the biodiversity of not just Peru, but the entire planet. So let's talk a little bit about that and how your research helps us to monitor the health of our planet.
Yeah, that's right. I mean, like a lot of entomologists that are in the kind of the specialty that I'm in, we both name and describe species and then ask bigger questions about the origins of a group, when it originated, where it originated. Praying mantises, for example, originated 200 million years ago. So they've been around for a really, really long time. And their pattern of dispersal across the world has a really specific type of pattern that you can detect. And we do that with molecular evidence and morphological evidence, but ultimately that gives us just a bigger picture of how a group that lives on Earth came to be. But the other shocking thing about it is that we only have described 1.3 million species of animals and plants on this Earth and bacteria on this Earth. So there's only 1.3 million described, and estimates have it around 10 million. So we're only 10 to 20% of the way there, even having a basic understanding of the biodiversity that's around us. And that's really fundamentally important, because if you don't know what's out there, it's really hard to ask questions about, well, what does it do? Is there environmental change? Is something becoming better, or is an environment becoming worse? So it's really a species is this basic measure of biodiversity. And if you don't have a concept of what that is, it's very, very hard to ask questions that apply to us as humans and see where we fit into the broader picture, but then just to get a general handle on what's out there.
While you were explaining that, our buddy Adam, who watches a lot of our Facebook feeds, hi Adam, thanks for being here today, was intrigued with the light trap, and I think he may want to try that out himself. So if he wants to investigate Ohio diversity in insects, how can he make his own light trap?
Well, it's been a specialty of mine as an entomologist. A lot of us become engineers because we're trying to get better and better ways of trapping our insects. Because if I go out into a rainforest and I just generally search, I can find a lot of praying mantises, but what's the most efficient way is to set up a light trap at night and then have them attracted and then come to you. But that only catches about 2/3 of praying mantises that are in a particular environment. So you have to kind of do both. You have to work constantly. But light trapping is an excellent way to observe biodiversity. A lot of people have porch lights or in really good locations, but you can build a light trap yourself and either run it on a small generator, or you can even plug it into a cigarette lighter in your car, even though most cars, I think, are doing away with cigarette lighters, I think. So that's becoming a gone technology. But you need to find a light source that is emitting full spectrum or ultraviolet light. And you can do this with a mercury vapor lamp. You can do this with a metal halide lamp. And you can also just buy a full spectrum LED. And a lot of people are actually converting to that because you can run them off of a battery and you can shine them against a sheet. And the reason why you shine them against a sheet is it amplifies the signal. So when an insect sees it, they don't see a point source of light, but they see a whole broad surface of light that allows them to come to it, settle on it, and then hang out. But once they arrive, most insects kind of become docile or inactive probably because they're nocturnal insects, and all of a sudden they're under light and they think it's daytime at that point, and they just settle in and wait for it to become night again. So it's a great way of observing stuff, but you can source lights really easily on a different light source online resale outlets, but look for that ultraviolet spectrum, and that's going to be the most important. And it's not ultraviolet to the point where it's going to harm you, because you got to get really, really ultraviolet to start causing big problems. It's in the probably the 370 to 400 nanometer spectrum.
I think you've just explained. No, that was good. Because now our social media guru Samantha is saying, tell Adam to send us pictures if he sets one up, so we can see how he's doing. I think you've just explained a phenomenon that I observed as a kid when we would go out at night, that there would be a cloud of insects around one light, like maybe the one hanging over the park next door, but then there'd be none over here by the lights by the parking lot. And it has to do with that UV spectrum, doesn't it?
That's right. And there's no light that works for every insect either. I mean, insects are queuing into very, very different sources. They're so diverse. That's like the key word of today, right? But every one of them is doing something a little bit different in the environment. So you see a mantis there, and then there's a katydid on one of the sides, and there's moths. There's a katydid. You can see it, the green one. They're all queuing into a little bit slightly different spectrum of light. You can get water beetles that will fly, and they'll only respond to a light source if it's putting out polarized light, because they're looking for light that's essentially just coming off of the surface of water, which is really neat because they're trying to find water surfaces. That's why you find a lot of aquatic insects that bonk into cars, parked cars, because the moonlight reflecting off the surface of the car, the glass, looks like the surface of water. So they land on it and get a rude awakening when they land, because they don't hit water, but they hit the surface of your car or your window. So it just depends on the insect. A lot of insects can come to a warmer light source, which is like a higher temperature. You can see this when you go to a hardware store. You can see light temperatures, like warm white or yellow light is like 3000 Kelvin. And you can go all the way up to 10,000, which if you put those light bulbs in your house, it's very, very white. But those sometimes are really effective for attracting insects.
The light that you're using on the trap we've been viewing, obviously you've hit upon a spectrum that is working really well for a large diversity of insects.
Yes.
And Carissa's a little concerned, I think, for your safety, because she wrote, when you're at the light trap, are the insects crawling all over you as well as on the sheet?
Yes, they're definitely crawling all over you. You have to go in and then work a little bit and then like get out when it becomes too intense. But you do get used to it. You don't get used to it when they're crawling into your eyes or they're going into your ear canals. So a lot of us carry earplugs or cotton in the field with us and kind of like stuff our ears full of cotton so we don't get stuff crawling in there. But it's like old hat to me. It's fine. I'm okay with it.
You may have just discouraged some of our viewers. Sorry, folks. Get them all out. We've got here we go. And I think Jim's picking up on that. Jim says many people write off these invertebrates as gross or scary. So how do you effectively educate people and break down these biases about all of these six-legged scientists that are out there?
Yeah, that's a challenge. It's always been a challenge. I think there's, like all organisms, there's a certain level, a certain threshold you have to get past. And I've really enjoyed working with praying mantises because they're an insect that kind of disarms people's gross factor with a lot of insects. Because they're one of the only insects that looks back at you when you look at it, and you really kind of think it's thinking about you and sizing you up to some degree. But I think it's kind of like one of these gateway organisms where you can start talking about the importance of these groups, because insects do $57 billion of ecosystem services for humans every single year. And that can be through waste disposal, through pollination, through many, many different things that they do in the environment. But their role is massive. And they might not be seen or they might not be heard all that much, but they're out there. And if they're not out there, then we start losing very, very, very vast critical features of our environment. And I always say, if you care about birds, you should care about insects. If you care about food, you should care about insects. If you care about most things, you should care about insects, because they are this fundamental part of our ecosystem. And without them, I don't know. I wouldn't want a world without insects because I love them, but I think we would suffer a lot if we didn't have them.
I would not want a world without food, so we'll partner up here and
Yeah.
save the planet.
Yeah, it's one of those basic things. Like, you have to have food, so you need to have things that are pollinating your food. And that's the other thing. Native bees are a significant portion of our pollinators, and also flies, and also beetles. And there's even some mammals that are pollinators. So when we talk about World Bee Day, which I think was actually just this past week, it's not just about the honeybees. They're a part of it, certainly. They're a very important part of it for our food supply, but the rest of plants are pollinated by both honeybees and our native insects and other animals that actually fulfill that role. And without it, our trees would suffer, and our wildflowers would suffer, and a lot of other things would suffer.
Very true, very true. You may have just inherited a new intern, because Adam's wife has just said if he tries to build a light trap, she's just going to send him over to work with you instead.
Tell him to send me an email, and I'll send him some specs on what he needs to build specifically. But then the most important part, if he plugs it into his house, to run an extension cord far enough away where everybody can be happy, right? It's not right on the porch or right next to the house, or right out a little further away.
There you go, Adam. We got you the hookup, man. Okay, let's get back to that little mantis, because it's got a name that I've been practicing. I'm probably going to mess it up now, but here I go. Vespa mantoidea. Is it Whirlii?
Whirlii. It's actually named. Oh. It's named for one of our volunteers at the museum, Rick Whirley, who works with us extensively on all of our kind of tech issues that we have in invertebrate zoology, and he does a lot of our imaging. So many of our papers that we've published recently have had images that have been taken by one of our museum volunteers, Rick. So he's taken on such an important role in our lab that it was something we really wanted to do to honor his help that he's given us, as to name a species after him. And this is a heck of a species to be named after. So it's a neat one. But yeah, it's Vespa mantoidea. There's a whole lineage of these little guys called Mantoididae, and it's a very, very early evolving lineage of mantis that lives in Central and South America. And the vast majority of them have been classified under one genus, Mantoida. This one was quite different from that, as I was talking about in the video. It's that wasp mimicry and that conspicuousness that made it very, very unique. So Vespa is wasp, and then Mantoida is the wasp Mantoida, to kind of capture that morphology and that behavior.
What is that process of name? Because there's one of my favorite things to do is look up interesting species names, and there, I mean, I can go down the rabbit hole of looking up Star Wars type names, because oh my gosh, there's critters named Yoda and Darth, and there's even one called the gene or the species name is Big Lebowski. So what is the process of,
Yeah.
I mean, you chose this one for your wonderful assistant there, but what's the general process of naming a new species?
Yeah, so I've been fortunate to name a lot of new praying mantis species, and Dr. Gunter, the other curator in invertebrate zoology, has named a lot of dung beetle species. For the most part, we pick names based on kind of what they might look like. They might have a special morphological feature, or they might have a special behavior. It's something descriptive, but oftentimes we'll also name them for the place that they were found, or maybe the person that they were found by, or just honoring a person that's important to you in science or some other aspect of life. So there's a commission of people and a rule set that governs the process, and it's under the International Commission of Zoological Nomenclature. And it's a number of scientists that are members of this, and they basically set the rules that we all follow. And what that does is it keeps everything standardized. There's a lot less chaos. There wasn't a commission early in the process of naming the life on Earth, and there's a lot of confusion because of that. Because sometimes something was named the same name as something else. So if you want to name something based on its green characteristic, there's only so many words describing green, so you might come up with the same exact name. So now you can only name something if the name is available, if the name is like not named by something else. And then it has to follow a specific rule set. You need to come back and you need to publish it. So you have to publish it in a scientific peer-reviewed journal, which means you have to have all the evidence necessary to justify that it is a species. You have to include or basically cover what that evidence is. It might be measurement data, it might be genetic data, it might be behavioral data. All of that together gives you what is a hypothesis of what a species actually is. And then other scientists need to agree, and then it needs to be published, and it needs to fit this whole rule set. So it's a scientific process with a specific methodology, and it takes quite some time, because if you want to name a new species, you have to review what is already known in that group. So maybe you find something in northern Peru or maybe southern Africa, and you bring it back, and you have to look at it and say, I think this is new, but I have to make sure it is new by comparing it with all the rest of the stuff in that group to make sure it is legitimately new. Otherwise, you run the risk of describing something that's already described, or maybe describing it in the wrong group from the get-go. So you have to do your homework.
It's good to know that committee does have a sense of humor, because I would encourage our viewers, not this afternoon, it's nice outside right now, go outside and have some fun, look for some insects. But when you have a rainy afternoon, just have fun looking up interesting species names, because there are a lot of them that are pretty hilarious. So that's good to know. There's a sense of humor in entomology.
Yeah, a lot of scientists like to talk about their other interests when it comes to that. So you get, like you mentioned, the Star Wars one, and I think there's actually a Beyonce fly, which was named by a fly biologist from Australia. It's pretty good.
Really? That's pretty good. Because Sage was asking if you had discovered another species, and I kind of want to answer that, because Sage, one of my favorite moments with Dr. Svenson was I did ask him, and now this was at least a year ago, I asked him how many species he had named, and he sort of paused for a minute and went, I'm not sure.
Yeah, I'm around 30 or so, I think, but it depends on your group, right? Like some mantises aren't that diverse compared to other organisms. So there's only 2,500 species in that order. And some other people might have 2,000 species within just their one little lineage. So beetles, for example, there's 400,000 species of beetles on Earth. So if you're a beetle biologist and you're working on a really speciose group, you might name hundreds and hundreds during your career. Mantises, it's not as much, but I think the cool factor of a mantis is brings it up a few notches. But I've named species after a few famous people. One of them is the wild krats. I named it after Chris and Martin Kratt, because I think their show is amazing. I've learned a lot from it, and my kids love the show too. This is it right here, actually. This is the wild crab mantis. So this was coincidentally, this mantis was found on the same exact trip that in northern Peru on the Amazon that we found the Vespa mantoida whirlii. And this one, unlike the bright orange, really characteristic pattern that we saw in the other one, this one looks like all the other ones in the genus and is very similar. And it wasn't until I got it home and did that comparison with all the existing knowledge that we had in the group that I did realize that it was a new species to science.
Man, that was a eureka trip. If you got 2 new species out of it, cool.
Oh, there's more. Lee, there's more. There's we have cabinets full of new species. Here's a fun fact, is most new species of mammal are actually described from stuff that's already in a museum, that's already been collected. It might have been collected for 100 years and it's been locked away in a cabinet just because the amount of time that it takes for people to actually get to do this stuff and do the research necessary, there's a huge lag time by the time when something is found in the field to when it's finally described. And that's one of the important things of why museums function as these big repositories of this really important information. Because at that preservation, if we didn't preserve stuff, we wouldn't be discovering as much as we would, because we're discovering stuff that we found 150 years ago, which is amazing to think about.
I got one of my favorite quotes from Roberta Mullenheim when I interviewed her, and she said, museum collections are a library of species. I just loved it.
That's right.
It's a great quote. And speaking of these collections, Craig would like to know, can you say something about the Mantis collections if they are available to view by the public?
Well, they're back in the research wing of the museum. So most people that visit the Cleveland Museum of Natural History don't realize that we have 2 levels down that contain all of our scientific labs and our collection spaces. If you are a member of the museum, twice a year, there's a members behind the scenes tour night that you can come back into the labs, and we display a lot of that material, and the praying mantis material is a part of that collection. And we pull all sorts of material out and show people directly when they come in and they have the interest to see more specific things. But in the collection right now, it's one of the largest collections in the world, and we actually have the Smithsonian collection of mantises in our basement right now as well. So there's, I think we calculated just about 18,000 specimens of praying mantis in the basement of the museum right now. So it would take you a while to see it all.
Right. I think you've also just won the award for most stealthy plug for museum membership. Well done. Oh darn! Did I just?
It does have its benefits.
You've intrigued Lance, and now he's wondering about your research and what it teaches us about evolution. And his specific phrase is linear, nonlinear. How would you define those terms within evolution?
Yeah, so I mean, I guess I'm a phylogeneticist, and ancestry and relationship are all basically tracked as this evolutionary trajectory kind of thing. So a lineage doesn't, I mean, in the fossil record, you might see evidence of change, gradual change from one organism kind of morphing gradually into another, which would denote kind of a linear evolutionary framework. But that's not actually how it works. Speciation and lineage formation happens by a branching pattern. So it's like kind of the branching pattern of a tree would be the easiest way to describe it, where you have one lineage that branches off earlier and the other lineage branches off. But these 2 groups might overlap for a certain period of time. So this one doesn't morph into this other branch. They simply share a common ancestor deeper in time that was the progenitor of those 2 branches in the evolutionary tree. So when you think about it in that terms, you might see a tree that has many, many branches lining up. You might have one organism here and another organism here and many in between that appear to have this gradual change from one to another. But evolutionarily, it didn't work like that. It worked through these series of ancestors that tracked all the way up to the most derived or the most latest branching groups within the family tree.
And then
chalkboard.
humans sometimes, yeah, where's the dry erase board? Exactly. Humans sometimes might accidentally mess around with this process because then we transport species into new areas. Adam was concerned about invasive species, and he was asking when you bring these new mantises back to the United States, they would be an invasive species. So you're not bringing them back alive, correct?
No, we're not. Anything that we do is permitted, so we have to have permits. We work with all the local governments and research organizations in the countries that we do our field work in, and we're given permits because we're sharing all that information with them. Most of the time, we're actually collaborating with scientists that are in the countries that we're working with. And then when we bring the material back, it's brought back preserved because we're interested in the morphology and the genetics. So we're taking a subsample of what we see in the country where we're doing work, and we bring that material back. Other people do need to bring live stuff back, and they are permitted by the U.S. Department of Agriculture and U.S. Fish and Wildlife, and have to have site inspections. Their facility has to meet a certain number of standards. But every so often stuff gets out, accidents happen. We saw that with, we see that all the time. I mean, there's species that get accidentally introduced or escape into different regions all the time. We're seeing that with this thing called the murder hornet, right? It's something in the Pacific Northwest. That is an escape, a documented escape. It's not established for what we know, and it's certainly not invasive because it needs to become like pervasive in an environment and then cause a certain threat. But as far as we know, that's not. But that is always a risk that scientists run when they need to work on living material and bring that living material back. But we don't do that at the museum. We're bringing back preserved material to work in museum labs.
I have seen at least one YouTube video of a very large green praying mantis munching on the head of a murder hornet. So I guess your vote would be if it was a smackdown, the mantis wins over the murder hornet.
I saw that too. I saw that same video, and I'm not surprised by it. The biology of a mantis would explain such a thing. They're opportunistic feeders for the most part. They're finding prey based on their level of hunger and their position in the environment and kind of what they're interested in hunting. So every mantis has a kind of a different specialty set of what they're interested in hunting or what they're capable of hunting. But ultimately, it comes down to their hunger level. And if they're hungry enough, they'll certainly catch much larger prey and much more dangerous prey. But for a mantis, catching a hornet like that, that's no big deal, because if they catch it in a way that it can't sting them, then the hornet doesn't have any defenses against the mantis. So it's sort of like I like the X-Men, right? Because there's always a mutation, but somebody else has a mutation that can overcome your mutation. You're never the top dog. There's always something out there that can get to you. That's sort of like nature. There's always something.
True, and hunger is a powerful motivator. And hunger, since we have now kept our, isn't it though, man? And it's totally after lunchtime, and we appreciate our audience sticking with us through the Facebook shenanigans. So thanks, folks, for hanging out. And gosh, now I'm all motivated to get out there and look for insects. So I think we'll wrap it up at this point to let you go escape and have a good Saturday. Dr. Svenson, thank you so much for taking the time to hang out with us this afternoon.
Yeah, thanks, Lee. It's been a pleasure. I'm really passionate about insects. I think they're critically important. I encourage people all the time to get out and look at them. There's all sorts of really neat ones that you can handle. There's stuff that you should just step back and observe, but they do play such an important role in our world that I think protecting them is a worthy conversation at any point. And I think if you do want to see them and you do want to come into the museum, there's all sorts of ways that we do invite people into the labs, and being a member is one of them. Signing up for camps is another. So there's ways of doing it, and that's what we're here for. We're here to tell you about what we do and how we do it.
And we love doing it too, doggone it. It's awesome. So thanks so much, folks, for hanging out with us this fine Saturday. And get out there and observe nature, man. It's going to be a fantastic weekend for that. Thank you again to Dr. Svenson. Again, my name's Lee. Thank you so much for joining us from the Natural History Museum at home. Head over to our website and check out more CMNH at home activities. Thanks, folks. Have a great rest of your weekend.